An aerial view of a city with buildings and trees, framed by green foliage. A scientist in a lab coat works at a counter next to a refrigerator, with biohazard signs on a door. A scientist in a lab coat and hood works inside a biosafety cabinet. A gloved hand removes a container from a liquid nitrogen freezer, creating a cloud of vapor.
It sounds like something that could be from science fiction.
A gloved hand holds a rack of small vials, covered in frost.
These are immortalized cells.
Two scientists in lab coats and gloves place a multi-well plate onto a microscope stage. A gloved hand adjusts settings on a tablet displaying a microscopic image of numerous small, circular structures.
Here we can see many different mini-tumors growing in this dish. This is a really robust case, and we know that it's resistant to a lot of therapies, so we're interested to find new treatments.
A frosted glass door with a nameplate reading, Office Jennifer Rosenbluth, and a woman working at a desk inside. Jennifer Rosenbluth, a woman with dark hair, speaks directly to the camera in a lab setting. A lower third identifies her as Jennifer Rosenbluth, MD, PhD, breast medical oncologist, associate professor, UCSF School of Medicine.
I'm Jennifer Rosenbluth. I'm a breast medical oncologist. I treat breast cancer patients. I specialize in breast cancer, and I also run a lab that...
A scientist in a lab coat walks into a lab, passing a calendar and a room number, K210A.
Utilizes organoid cultures to model breast cancer and breast cancer risk.
A top-down view of a scientist working inside a biosafety cabinet with various lab tools. A gloved hand points to a microscopic image of pink-stained cells on a tablet. 3 microscopic images of intestinal organoids are displayed. Text: Intestinal organoids, Bayer Lab. A colorful microscopic image of a brain organoid. Text: Brain organoid, Kriegstein Lab.
A lot of people think of organoids as mini organs, or in our case, mini tumors that are grown in a dish. And
microscopic images of breast tumor organoids glowing in various colors. Text: Breast tumor organoids, Rosenbluth Lab. Jennifer Rosenbluth speaks to the camera in a lab.
And that is true in a sense, but they're really a simplified system.
Several dark, circular brain organoids under a microscope. Text: Brain organoids. A multi-well plate sits on a microscope stage illuminated with blue light. A female scientist with red braided hair looks up and speaks.
They're alive and growing well. What's cool about these ones is that they're also fluorescent.
Microscopic images of breast tumor organoids, some glowing brightly. Text: Breast tumor organoids, Rosenbluth Lab. 2 female scientists in lab coats stand next to a microscope, looking at a tablet. A gloved hand touches a tablet screen, displaying a microscopic view of cells on a microscope stage.
You can generate some organoids using a type of stem cell technology.
A close-up of a tablet screen showing a microscopic view of a dark, irregular organoid and adjusting settings.
Or you can generate organoids from a tiny piece of a normal tissue or a tumor tissue.
You can see like the nucleus.
A gloved hand adjusts settings on a tablet showing a pink-stained tissue sample under a microscope. A microscopic view of pink-stained tissue with a circular structure.
They're a way for us to take tumor material from a specific patient's.
A close-up of a multi-well plate with handwritten labels and some wells containing samples.
Tumor, grow it in a lab, make miniaturized versions that we can then use to.
A male scientist in a lab coat and mask walks out of a lab room, holding a multi-well plate.
Do things like test drugs, model, study in real time now in a laboratory dish.
A male scientist in a lab coat and mask pipettes liquid into a multi-well plate inside a biosafety cabinet. Jennifer Rosenbluth speaks to the camera.
As I see in the clinic every day, patients with tumors of the same subtype and same features can respond very differently to the same drug. And now we have a model system that really lets us study that in the laboratory.
A gloved hand removes a large, round lid from a liquid nitrogen freezer, releasing a large cloud of vapor. Takes a multi-well plate from a shelf inside an incubator filled with many other plates, and holds a multi-well plate with samples and handwritten labels.
Once an organoid is established, we can replicate.
A gloved hand scrolls through microscopic images on a tablet screen.
That tumor and make many mini-tumors, and now we have sufficient numbers to test even large number of drugs.
2 female scientists, one in a lab coat and one in a blue top, look at a laptop screen and smile. A blurred image of a scientist writing with a white text box overlay. Source: BioRxiv, Organoid-evaluable clinical biomarkers predict drug responses and guide new breast cancer therapies.
In this study, we identified that most clinical biomarkers
The white text box expands to include a list of authors and a DOI link.
can actually be evaluated in organoids. So
Jennifer Rosenbluth speaks to the camera.
a biomarker that predicts response to a specific therapy also predicts response to that therapy in the organoid model.
A male scientist in a lab coat and gloves opens a refrigerator and takes out a vial. A gloved hand uses a pipette to transfer liquid into a small vial.
If you know there are 3 drugs that might work for that patient, now you can actually test those 3 drugs in a
A male scientist in a lab coat and gloves works with vials inside a biosafety cabinet.
miniature tumor model from that patient. And maybe you can say, hey, of those 3, this is the one that has an amazing effect.
Jennifer Rosenbluth speaks to the camera. A microscopic time-lapse shows numerous dark, circular tumor organoids, some appearing to shrink or disintegrate. Text: tumor organoids dying.
You can see the tumor cells dying as you give them therapy, so it's a very visual experience.
A female scientist points at a laptop screen showing a microscopic image of cells. A close-up of the laptop screen shows the microscopic image of cells.
You see already within 12 hours they start to fall apart, and that is because in this condition we added a drug that was very effective in killing these organoids.
The microscopic time-lapse continues, showing more tumor organoids dying, with a red circle highlighting a disintegrating cluster.
Before this experiment, Tambin could add really high concentrations of chemotherapy and it wouldn't touch them. They'd just be perfectly happy. So here she's identified something that's finally able to kill these really resistant tumors.
A female scientist in a lab coat and gloves works with a microscope and tablet at a lab bench.
There's 2 different kinds of research.
A close-up of a microscope lens and stage. A female scientist is blurred in the background. A lab coat with University of California embroidered on it hangs in the foreground, with 2 scientists working in the background.
One is discovery research, so finding new therapies that are going to work for many patients. And
A male scientist in a lab coat and headphones writes on a whiteboard inside a biosafety cabinet.
the other is the idea of personalized medicine, where you're testing a drug to identify the drug for that specific patient from whom that organoid is derived.
A gloved hand places a small vial into a vortex mixer. Another holds a small vial with yellow liquid, shaking it.
This is an experimental drug, and it's like pretty potent against cancer. The reason why we don't use it in people is because the moment it gets in low temperatures, it clumps up into this crystal at the bottom.
The male scientist shakes the vial, examining it closely.
So I'm trying to shake it to the point where the crystal goes away.
An exterior view of a modern UCSF building with people walking outside. An aerial view of the UCSF campus buildings under a clear sky.
UCSF is at the forefront for personalized breast cancer
Jennifer Rosenbluth speaks to the camera.
treatment through our large clinical trial programs, and now we're
A gloved hand points to a multi-well plate on a microscope stage, then removes it.
finally able to integrate organoids into that framework and hopefully improve our clinical predictions.
A close-up of a tablet screen showing a microscopic view of numerous green-tinged organoids, with settings controls on the side. The UCSF logo appears on a dark blue background.